At the end of the last lecture, I had a graph, the approximate geological history of CO2 concentration along the span of time that animals have been around on planet Earth, pointing out, among other things, that 10,000 years or so ago, until we started burning stuff willy-nilly, CO2 concentration in the atmosphere was lower than it has ever been in the history of animal life. If you extend this back further, it just keeps going up, because CO2 was one of the dominant gases in the Earth's atmosphere when it formed and when life originally arose. So, a billion years ago, 2 billion, 3 billion, so on. Now in red, I'm going to sketch on here approximately the history of oxygen concentration in the atmosphere. So if this is present day levels of oxygen, about 20% of the atmosphere, we think of that as good. We depend on oxygen. It's been about like that for a while, except for a little blip in the Carboniferous when it may have gotten up to 35% or so. 40 is when building things out of wood becomes a really bad idea. There's a couple of big changes. I haven't quite got them in exactly the right place, or the numbers exactly right, but the point I want to make is that at the dawn of life, the atmosphere of the early Earth had practically no oxygen in it, no molecular reactive oxygen. Sometime around here, what happened? In this zone, 2.5 to 2 billion years ago, some group of bacteria that we now call the cyanobacteria figured out how to harness the energy from light to capture CO2 into sugars, producing oxygen as a waste product. These original polluters caused the first big rise in atmospheric oxygen concentration. The reaction of photosynthesis, recall, is something like this. That's supposed to be a photon or something. CO2 and water going to make, excuse me, carbohydrate and oxygen. This is formaldehyde, incidentally, the simplest carbohydrate. Highly reactive, bad news, right? String a few of them together and you have a sugar. Glucose means putting 6 of those together. But this is the really bad news from the point of view of things that lived back here, okay? This oxygen reacts with practically everything. This highly reactive gas started to accumulate, and not only did it poison everything that was alive, then it also took all the iron, all the soluble iron, which used to be quite abundant, down to the bottom of the ocean, turned it into rocks. Nowadays, iron is scarce. I mean, you can mine it, but free iron floating around is a limiting nutrient, and we scavenge. All life forms today scavenge every iron atom that they have. That's, incidentally, why your blood is red, but your pee is yellow. You take the iron atoms out and save them before disposing of the easy-to-replace carbon chains that hold it in oxygen-carrying proteins. Okay, anyway, so this is the first great episode of climate change driven by organisms that don't care what they're spewing into the atmosphere. what's going on here? This should actually be shaped a little more gradually up to the Cambrian. So let's put it like that. Very likely, it's sometime around 1 billion years ago or so that the history of eukaryotic photosynthesis begins. So the prokaryotes started it back here. Cyanobacteria learned to conduct this reaction and make their own food out of sunlight and CO2 in the atmosphere. Eukaryotes initiated the next great phase in the history of photosynthesis when they teamed up with cyanobacteria rather than inventing photosynthesis on their own. So all these blue-green bacterial cells floating around, making excess sugars happily. Surely they were tasty. And the scenario goes roughly like this. Some green flagellate is normally in the business of eating these things, predating upon them, engulfing them, and digesting them and as food. Had the bright idea put in cell biological terms, this message is that some enterprising cell figured out that if it delayed digestion in lysosomes of these cells producing excess sugars, it could derive more benefit than by digesting them immediately. And so the imaginary scenario is that heterotrophic flagellates like these, heterotrophic mean they grow by eating other things, over evolutionary time, in effect, learned to maintain something like a cyanobacterium intracellularly as a symbiont. The flagellate descendants of such an enterprising eukaryote have chloroplasts with their light harvesting system and with the remnant genome of the original cyanobacterial symbiont partner still in there, and no further need for heterotrophy. And we would contrast this condition, photoautotrophy, with heterotrophy. This means self-growth light, right? There are certainly several steps along this transition from a predator-prey relationship to a stable endosymbiosis, in which, in fact, the descendants of the cyanobacteria that became chloroplasts, the chloroplasts of today's various algae, certainly can no longer live independently. They are integrated parts of these cells. They are integrated organelles within these cells. One of the steps that must have had to take place is to couple host and symbiont division. Today's algal and plant cells have anywhere from one to many, many chloroplasts. A cell with very many chloroplasts, of course, could just count on a few getting into one side and a few getting into the other side. It's not so simple for small cells like the kind of flagellate I've drawn here, which might have just one or two. There must be some mechanism to link the division of this now essential organelle. Another key step must have been to have mechanisms to feed and maintain symbionts. Delaying the digestion of a captured cell isn't all there is to it, and the trade for incipient symbioses of a similar kind might be to provide, for example, something that the bacterial cells need to survive in exchange for the excess the excess products of photosynthesis, the sugars. Third key step, there has to be some means to limit symbiont escape or degeneration. Key to all of these steps is the invention of some sort of transport path within this endosymbiotic cell to import cytoplasmically translated proteins across membranes enclosing the captured symbionts. In modern plants, modern algal cells, the chloroplast has a bilayer membrane enclosing it, plus internal membranes on which photosynthesis is conducted, typically arranged in stacks. So this is the chloroplast outer and inner membrane. These are so-called thylakoid membranes on which the light harvesting complexes are arranged. There's very often an object in here that looks rather distinctive. This is a feature in particular of many algal cells. This thing surrounded by starch deposits is called the pyrenoid. And that's the site of most of the actual CO2 fixation. Inside is a matrix where the enzyme rubisco that actually accomplishes CO2 fixation is concentrated, and membranes stretching in there from the thylakoid stacks, as it happens, deliver things, including bicarbonate ion, which is then converted locally by carbonic anhydrase into CO2 to feed the enzymes that actually mediate carbon fixation. All of this requires some sort of transport complex about which we will have no more to say in detail. Transport proteins must mediate traffic into and out of the chloroplast from the cytoplasm. Almost everything that's in here is transported through this gatekeeper, with the exception of the few products that are still encoded in the chloroplast's own genome. I mentioned in a previous lecture that there are 3 lineages today that are considered to be the descendants of this primary endosymbiotic establishment. That is, the Archaeplastida consist of the glaucophytes, the chlorophytes, the green algae that is, and the rhodophytes, the red algae. Of all these, the glaucophytes, which students taking the lab class will hopefully meet in person, look the most similar to this kind of imaginary intermediate stage. The glaucophytes still look like flagellates that swallowed a bacterial cell and didn't digest it. Indeed, the photosynthetic organelle called a cyanelle in there has a peptidoglycan cell wall still, like a free living bacterium would. They cannot, however, live independently. It's not as if you can just burst open a glaucophyte cell and have some cyanobacteria that you can keep in a petri dish no matter what you feed them, because a big part of the history in all of the plastid-bearing lineages is the transfer of genes from the original cyanobacterial genome to the eukaryotic nucleus. That is one great way to limit symbiont escape, of course, is make them dependent on you by capturing essential parts of their genome so that they can't live independently. The transfer of genes from one genome to another here necessitates, again, the acquisition by those individual genes, for the sake of their products, of targeting signals so that they can be recognized and transported by the machinery that imports things into the plastids. So this is what we would, the story we would tell roughly for primary endosymbiosis. Secondary endosymbiosis is the next step. And it is secondary endosymbioses that give rise to all of the other photosynthetic lineages, of which there are very many across the eukaryotic world, including the diatoms, the photosynthetic euglenids, various others. And the story there is that some green flagellate or rhodophyte, or perhaps glaucophyte, although nobody knows of an instance of such, with its ability to photosynthesize, is targeted by and eaten by some other eukaryotic flagellate. Now, this captured prey must be inside a food vacuole somewhere there. Look, there's its flagellae. How is the predator going to make use of this useful capacity to photosynthesize in this circumstance? In order to stably acquire photosynthetic capacity, somehow this plastid had to become the endogenous chloroplast of the once predator. What did that involve? Does this cell simply strip away all of the eukaryotic components and say, well, that's mine now, thanks for the batteries? No, it's not so simple. In fact, as we'll read in a couple of papers, in some existing groups, two existing groups, there remains a relic of the eukaryotic nucleus from the captured eukaryotic lineage. In others, the eukaryotic component has been stripped away. This cell, the secondary endosymbiont, must have had to acquire, again, likely by transfer of genes from the captured genome to its own genome, the capacity to maintain the chloroplast, the capacity to transport proteins into the chloroplast, and the capacity to synchronize its division, feed it appropriately, etc. This has led to a lot of, in the historical phycological literature, led to a lot of membrane counting. Like, well, okay, so there's a food vacuole here. Maybe that lysosomal membrane became the outer membrane of the chloroplast, right? And so then when one of these eukaryotic algae got eaten, there would have been a new lysosomal, a new food vacuole membrane added to that. And so perhaps the lineages in which there are multiple membranes surrounding the chloroplast, those membranes represent the evolutionary history of engulfment. Well, that's probably not true. Membranes are a little easier to rearrange than all that if these cells had wanted to do that. Nevertheless, it does illustrate the complexity of combining genomes and the necessary transport machinery. Then there are tertiary endosymbioses. And that simply means more of the same. That is, that some lineage acquired its plastids from not one of the primarily endosymbiotic groups, but from one of the secondarily endosymbiotic phototrophs. Examples of secondary endosymbioses that we'll read about include cryptophytes in particular. One of the groups where the nuclear genome of the captured eukaryote from which cryptophytes got their plastids originally remains in there tucked in there with the chloroplast. And then we will see examples of tertiary endosymbiosis in the dinoflagellates. I'm going to sketch out cell structure and some other features of a series of 6 flagellate groups now, briefly, starting with the prasinophytes, which means green plants, basically. This is a basal assemblage amongst the green algae. That is, they don't necessarily all belong together, but they look like they have traits that are primitive to the group to which they belong, and they provide some of the best extant models for what the primary endosymbiosis might have looked like once it was established. Many of them are fairly simple and round, but some of the more interestingly shaped ones are kind of apple-shaped or something like that. And have 4 equivalent flagellae, often long enough that they can beat them with a sinusoidal waveform. Of course, they have a plastid, often one or a couple that are sort of cup-shaped right under the surface of the cell. So that's supposed to be the chloroplast, and typically that includes a highly visible pyrenoid. In many prasinophytes, you'll also see somewhere red, because it's rich in carotenoids, eye spot. So that's what that is there. Of course, there has to be a nucleus in there somewhere. These cells very often have a vacuole, and stretching down from the basal bodies there'll be a contractile rootlet, a striated rootlet. Let's see, let's put the basal bodies in there. Of course, like any eukaryotic cell, they have their own endomembrane system with organizing vesicle traffic and secretion. And this reservoir here, this pit, is a reservoir for scales. Prasinophytes are typically coated with some kind of organic scale, not a definitive cell wall, but a covering of overlapping scales that are sometimes of intricate architecture. Okay, so key traits. Scales, no wall. They have an open mitosis, meaning that the nucleus breaks down as they go through mitosis. And they undergo cytokinesis by furrowing. Doesn't mean it's exactly the same mechanism as in animals, but the cell membrane pinches in circumferentially to divide the cell in 2. 4 or sometimes 8 equal flagella that typically emerge from a pit somewhere on the cell surface. And there are examples, we'll read about one, that are phagotrophic, that is, they eat. Phage is eat. This, of course, is of interest because the fact that they have the apparatus to conduct phagotrophy, to take in bacterial cells and digest them, validates the notion that perhaps once upon a time the ancestors of these cells lived that way, and that that's how they acquired their endosymbionts originally. Derived forms amongst these include Tetraselmis, which has an important contribution in the history of biology and also shows up in interesting ways, for example, as symbionts in flatworms, um, allowing some of the few green animals that are out there. Tetraselmis, as it happens, has fused its scales into a complete cell wall. Lab students will get to meet Tetraselmis. The chlorophytes, the next group we're going to talk about, probably arose from something not that different from Tetraselmis. Other interesting forms that you will hopefully meet in the lab if you're taking it are Peramomonas, which is actually what I'm drawing here. Cells related to Peramomonas, including Halosphere, which in lucky years I can show in the lab. Make, in addition to unicellular flagellates that swim around, they make giant cysts that appear in the plankton that are thought to likely represent a product of a sexual phase in their life cycle. These giant cysts then carve themselves up into many individual flagellates at some transition. All right, now the chlorophytes, and this word keeps getting applied to either the whole of the green algae, including plants, or just a subset of it, or whatever. The group I'm actually talking about is the chlorophycians. One letter distinction: phyte is plant, phyke is seaweed. Okay, so in contrast to the prasinophytes, which are probably the least derived, that is the most primitive amongst the green algae, the chlorophycians are amongst the most highly derived. This group includes, incidentally, Chlamydomonas, which you have heard about previously because of its important role in the history of studies of ciliary function and structure. In fact, the group to which Chlamydomonas belongs is probably amongst the most highly derived group of chlorophytes and includes multiple inventions of a multicellular life history. Chlamydomonas itself is unicellular, but some of the basal groups amongst chlorophycians are multicellular, and so that raises the possibility that Chlamydomonas actually might be secondarily unicellular, that is, that it had multicellular ancestors. And I'm going to draw Chlamydomonas as a contrast to the Peramomonas diagram over there. So Chlamydomonas, as you already know, is biflagellate, is very round. Two flagella, and there are basal bodies in there. And there's various apparatus connecting them. So, for example, they can change their swimming pattern by shifting the orientation of those things. unlike a typical prasinophyte, Chlamydomonas has a rigid cell wall. This is not just a cellulose assemblage like the cell wall of land plants. It's independently invented from that. This rigid cell wall confines cell shape and also limits the movement of things on the cell surface. Again, they've got a plastid in there, typically one large cup-shaped plastid. Incidentally, why prefer the term plastid to chloroplast? Simply because chloro means green. Not all plastids are green. Chlamydomonas also has a prominent eye spot that's red. We call this cell wall theta, typically. Again, that's the pyrenoid. What's the eye spot for? Well, as these cells swim, they rotate, and the eye spot combined with a photosensitive patch somewhere, as the cell rotates, the eye spot shades differentially this photosensor relative to some light source, some directional light source, like, say, the sun. And consequently, by modulating the beat rate of one of the flagellae relative to the other flagellum, the cell can turn and swim toward the light. Many Chlamydomonas and their relatives live in lakes or ponds, but many of them also live in damp soils. And so phototaxis is an important part of their life. Key traits. Again, they have a actual cell wall that's quite rigid. They have a closed mitosis. The nucleus does not break down during division. The nuclear membrane remains intact, mostly. Excuse me. Cytokinesis involves furrowing, but with an adaptation of this group called a phycoplast. During division, if there are poles to the spindle here and chromosomes lined up somewhere in here, as this divides in anaphase, some bunch of microtubules appear perpendicular to the spindle axis, prefiguring the future cleavage plane. As the nuclei reform, the spindle collapses, with this emerging wall of parallel microtubules separating the two daughter nuclei, and a cleavage furrow meets this so-called phycoplast, in which vesicles are fusing to create the partition between daughter cells. So that's yet another mechanism of division. Even a third mechanism is embodied in other members of the green algal supergroup, including the land plants. Finally, usually two equal flagella. I guess I gave up on trying to call them cilia instead of flagella. One of the things that I want to point out in passing is that the rigidity of Chlamydomonas cell wall imposes a so-called flagellation constraint on division. The basal bodies and the axonemes cannot move through this rigid cell wall, which again is not just made of cellulose like a plant cell wall. So cells have to shed their flagellae every time they want to divide and regrow them afterwards. So ciliary regeneration is a regular part of the life cycle, as I have also pointed out for animals. When animal cells want to divide for other reasons that remain mysterious, they always withdraw their cilia. Now, in the case of phototrophic flagellates that depend on the light, losing your cilia might be bad while you're trying to stay in the light by, say, swimming toward it. So as it happens, many representatives of this group that includes Chlamydomonas may delay division until nighttime and then go through multiple rounds of division within the mother cell's theca. So they could spend daylight accumulating resources through photosynthesis, and then do all their division at night when there's no point swimming up to stay in the light anyway. Sexuality in Chlamydomonas is interesting and similar in many related chlorophycians. In Chlamydomonas, one mating type, which is usually called the plus type. Here it is with its cell wall and a little protuberance, a little sticky outy part grows between the flagella. And a cell of the other mating type, the minus type. Again, here's its cell wall, its theca. It snuggles up. And they mate flagellum to flagellum, zippering them up together, gliding along each other's flagella, as it were. Now, these are haploids. They use their flagellar adhesion to snuggle right up close. And this actin-rich protuberance, this mating device on the plus type cells, fuses with a receptive spot on the minus type cell. And this diploid zygote, which will end up with 4 flagellae, at least briefly, hatches out of those original cell walls. we'll pick them up there a little later in a subsequent lecture.